I did? Oh goodness, this is falling apart on me. It's some research I conducted at Oak Ridge National Laboratory with Gary Van Berkel and Vilmos Curtez, where we determined concentrations of analytes and their metabolites, or drugs and their metabolites using electrospray mass spectrometry. So the drug development process is extremely expensive today. For a major pharmaceutical company, it costs them around $350 million and takes around 10 years to get a single drug from conception to market. And along each step of that process, hundreds of drugs will fail to make it past a given step in that testing. And so the total drug cost to a major pharmaceutical company averages out to around $5 billion. This is from 2013. For each successful drug that actually does make it to market. And so when they then give that drug to consumers, they have to try and make up all that money that they spent in research and development, so they simply pass that cost on to you. So one of the key goals for researchers today is to discover ways that we can reduce the cost of the drug development process. The drug development process, as a matter of fact, has quite a few different steps, more than just these, but I've sort of simply broken it down into a couple major ones here. So we have synthesis of new drugs, screening those drugs in what we would call in vitro studies, where we look at how the drug behaves in regards to the disease or how it reacts to individual cells. We look at animal testing, see how drugs interact with organisms that sort of model humans. And then if that's successful, we then go on to clinical trials where we actually test it in patients. And then hopefully at the very end of this ordeal, you'll manage to get FDA approval and see your drug on the market. But every single one of these steps has a possibility for seeing improvement in efficiency and cost. But at every single step, what we have to make sure we maintain is that even when we try to reduce costs, we can't cut any corners in terms of how well we test the drug's efficacy and its safety. What I'm going to be focusing on today is strictly the animal testing phase and examining ways we can improve this step. Animal testing has an extremely long history in drug development, starting with IG Farben in 1930s when they developed Pronicil, which was the very first antibiotic ever made. When they tested this, what was originally a dye compound, just on bacteria in a dish, it had absolutely no effect. However, when they tested it in mice that were already infected with bacteria, the mice were cured, which was the first at the time. And the reason for this is because this molecule shown here is actually cleaved at this middle bond. And what that results in is that this left side here is a very potent antibacterial. And the key concept I want you to take away from this is simply that when drugs enter the body, they can sometimes change. And it's important for us to examine how they interact within the body, because simply using cellular tests can often fail to detect what exactly the full picture of the drug is. So when researchers sort of look at the full picture of how a drug behaves in your body, they typically look at what they call ADME, the absorption, distribution, metabolism, and excretion of the drug. Absorption, of course, is simply how the drug gets into your body. This can occur in a variety of different ways, whether it's oral ingestion, or you can use inhalation, injection, or even topical creams. And a key component that I want to drive home on this topic is that properties of both the drug and the disease that it's treating will dictate a large part of how you choose to absorb the drug. So for instance, if a pill struggles to handle strong acids, if it breaks down too easily, then using it in an oral pill simply will not work. And on the other hand, here we have two examples of drugs where the disease has dictated how we choose to deliver them. So in the case of asthma, and in the case of, say, an allergic reaction, you want to get those drugs into the body immediately. You can't wait an hour for the pill to take effect. So we use either inhalation directly into the lungs, where you're having an asthma attack, or sending epinephrine directly into the bloodstream to sort of treat those drugs at a much faster rate. Once a drug is into the body, it then is transported through the bloodstream and goes into the organs where it does its work. However, there's a large part of the distribution of the drug where you want to tell where it goes and how long it stays there. So some critical examples here include the blood-brain barrier, which typically can block certain compounds from crossing over into your brain, which is normally very helpful. But if you have a disease that's infecting the brain, you want to make sure that whatever drug you've designed is able to cross that barrier and do its treatment properly once it is across there. You also want to examine how effectively drugs are stored in fat. because if they are stored in fat, they can bioaccumulate within your fat cells and then build up to a higher and higher concentration, so that even if what you initially had as a drug was not very toxic, if it bioaccumulates at high levels, that will create a potentially dangerous and harmful situation. Metabolism of drugs is how you actually remove drugs from the body. It's not so much the metabolism that you often hear about, how much can you eat without gaining weight. It's very rigidly focused in the chemical sense of how it's broken down and transformed in order to make it less reactive and more excretable. Okay, this is done almost entirely in our liver with two different sets of steps. One is phase 1. In phase 1, we usually add or remove oxygens in order to make the compound more water soluble. And then phase 2 usually involves transferase enzymes, which will then add on special conjugate groups such as sugars or amino acids to make sure that the drug is even more excretable than before. Excretion is obviously just getting the drug out of the body, however that happens. So you can do that with urine or stool, through renal or biliary excretion. And then there's also exhalation and sweat are some less common ways that some drugs can be removed. And one key component of this is that you can remove both the drug directly without metabolism, or you can require, some drugs will require the metabolites in order to actually be pulled out of the body. So taken all together, these, the absorption, distribution, metabolism, and excretion have a profound impact on how pharmacists choose to dose drugs that you take. So depending on how quickly it's absorbed and excreted will dictate essentially whether or not you need to take another dose, how large that dose you take is, and then whether or not it's going to be harmful to put you on that medication for a very long time. So if you, in fact, if you have problems with any of these functions, for instance, a failure to metabolize properly or a failure to excrete properly, these are usually what cause side effects, and they're also what can even cause fatal problems with drug complications. So in this case here, I have an example of sort of a hypothetical situation of how drugs temporally can interact in the body. So on the x-axis here, we see time after the initial dose. And on the y-axis here, we have bioavailability, which is a shorthand term for concentration in the blood. This red horizontal line here is the minimum effective concentration. Drugs below this concentration are simply not going to do any treatment. Up here in green, we have the maximum tolerable concentration. Drugs which exceed this level begin to cause damage within your organs. So here in the blue line, I've depicted one example of a drug that would fail. In this case, this drug does begin treatment very quickly, but it actually exceeds the maximum tolerable concentration, begins to damage, and then it crashes out at about the 3-hour mark, which means if you were to try and design a drug, or design this as a treatment, you would have to retreat yourself every 3 hours and re-expose yourself to a harmful level of the drug every 3 hours. The second drug here depicted in pink has sort of the opposite effect. It takes 5 hours for the drug even to begin to treat the disease in question, and then it is stored in your body and not removed. So in both of these cases, which are very different, we see the need to understand what this drug is doing when it's in your body, or else we have no idea whether it's not safe. So in order to even begin to get this drug, in any example, into a human subject, we want to first make sure we understand very completely that it doesn't do either of these examples or any other sort of aberrant sort of metabolic pathway to see if it's safe. Now the way we typically do a metabolic study is we'll take a number of mice or other mammals, we'll dose them with a certain specified amount of drug, and then we will one by one sacrifice the mice at differential time periods in order to understand how the drug varies over time. The mice are then excised and their organs are removed. You have to blend the organs up and filter them through a column. It's a pretty unpleasant process. I didn't have to do this part, thank God. And then you have to measure the drug and metabolite concentrations at the very far end. My entire research goal during my summer at Oak Ridge National Laboratory was simply to find a way to use a method developed in the lab that I worked in to speed up the process. We wanted to eliminate the column extraction, eliminate the surgery, and eliminate the need to blend the organs up. What we did instead is we would freeze the mice and put them through a miniature ham slicer, essentially, to sort of like make really micro-thin sections of about 1 millimeter thick. And so what you see here is a mouse. Here's the head, and this end over here is where the back end would be. And we would mount this onto a glass slide and after it was flash frozen. And then we would examine each of these different organs, and our goal is to simply use a surface analysis test where we have a small amount of solvent placed onto a spot and have the solvent pick up the analyte and then add that to our analysis technique. I'd also like to mention at this point that one of the key ways where we're going to be actually measuring concentrations is we're going to be using blood spots here. So these were blood spots without any drug in them that was then artificially added to create what would be a standard curve to sort of measure an actual amount of analyte. Coming out the far end. So here is the actual liquid extraction surface analysis technique, which was developed at the laboratory I was in, which was the Organic and Biological Mass Spectrometry group. What we do is we have a robotic arm which selects a new sampling tip for each sample, draws up a specific amount of solvent, approximately 100 microliters. So then it moves over to the spot in question, which we specify using a computer program, and it will automatically dispense the solvent onto that sample, wait a certain amount of time at the surface, draw the sample back up, and then inject it into the mass spectrometer. To look very briefly at what this looks like in the actual lab, this is a very small box about that big. You have the robotic arm up here, solvent wells hidden in the back, a wide array of tips, and then your tissue slide location. And then over here, you have your mass spectrometer, which is our analysis technique. Speaking about mass spectrometry, it was developed 100 years ago last year by J.J. Thomson, a British physicist, who also is credited with inventing the electron, or not inventing, discovering. Didn't invent it. No, he did not. What it's used to do is you measure the mass to charge ratio of specific ions in the gas phase. So on this chart here, you can see mass to charge ratio labeled on the bottom, increasing left to right. And individual peaks of very specific height are shown for ubiquitin, which is a protein. And this protein has multiple can have multiple charges on it. And so as you increase the charge, the mass to charge ratio is then decreasing. So what you want to do in order to quantify something, because this gives you both quantifiable data about how much of the analyte you have, and it gives you characterizationable data in terms of how big is the analyte in question, how much mass does it have. So it requires you to have three things. You need a mass. Well, that's great. That describes everything on the periodic table. That's a great place to start. But it has to be charged, and it has to be a gas. And this is actually a considerable problem for quite a long time. Now, mass spectrometry could analyze compounds like the one on the left for since about the 30s or 40s pretty reliably. However, for compounds on the right, which is a protein in this case, it's so much larger that to get into the gas phase, you can't simply use heat to sort of evaporate it and then ionize it in the machine in the same manner. So for a long time, this was a barrier to doing biological molecule analysis. And so, however, in the 1980s, methods were developed for actually characterizing and analyzing these compounds. So the 2002 Nobel Prize in Chemistry, Kirk Wutrick, which I'm sure I butchered, won for NMR analysis, and then both Koichi Tanaka for MALDI mass spectrometry, which is a technique I'm not going to be talking about, but then John Fenn for electrospray mass spectrometry. All three shared the prize for studying analysis techniques for biomolecules. Electrospray, which is the technique that I worked with 2 summers ago, and is in fact the technique we have downstairs in our basement on our mass spectrometer, focuses on using the natural evaporation of solvent molecules to get things into the gas phase. So if you go back, think back to that initial picture of my schematic, the robot and such, this would be the solvent tip. Here would be fluid with our solution in it. On this end is the mass spectrometer, we'll get to in a minute. And what we have here is we have a large voltage difference between these two. So that way positive charge builds up here at the tip and is eventually transported in tiny solvent droplets over to the plate on the other side. We have a sheath gas here, which is going to make sure that we have dry analyte actually reaching the mass spectrometer. But the key component is what actually happens if we zoom in here on the evaporation of solvent molecules. So you have a tiny droplet of solvent. As it gradually evaporates away, all that's left behind is a bunch of charges that are stuck to your analyte in question. And so whether that's a protein or another sort of molecule, any of those will do the trick just fine. How do we actually measure things in a mass spectrometer? Well, in the case of the lab I was working in, we use what's called a triple quad mass spec, which means it has 3 quadrupoles. Very nice name. What we do is we begin with a lot of different analytes, so we don't have to do any filtration. This first quadrupole, what it is it's an electric field that we can oscillate back and forth. And based on specifying the frequency of the oscillation of the electric field, a certain size mass to charge ratio will pass through, but other size particles or other charged particles really will fall out of that path and be able to make a straight line through. In the second chamber here, we have an inert gas which will fragment our compound of interest into tinier pieces. This is really important if you want to characterize what your analyte looks like. If you're not entirely sure what its different fragments, what it is, by looking at the different fragments and pieces, you can sort of piece together what the full picture is. And then at the very far side, this third quadrupole is generally for separation of, if you want to look at a specific fragment and quantify it. But on the far side, we have a detector that will count how many ions are reaching the far side. And by doing that all together, we can get a quantitative idea of how much analyte we had in our initial sample coming in. Putting all this together, we can use this to detect drugs. So in the case of the research I did, we used 5 drugs. Here we have verapamil, which is a calcium channel blocker. We have propranolol, a beta blocker, which we'll talk about more in a minute. Diclofenac, which is an anti-inflammatory. Acetaminophen, which you recognize as Tylenol. And terfenadine, which is an antihistamine or allergy medication. The key component I want you to get from this slide is that these are old drugs. None of these are new. And so the data about their metabolism and where they go in your body is extremely well known. We've known it for years and years and years. And that's important when we're trying to confirm our method and to see how well we can measure drug metabolism. Today, I'm only going to really have time to focus in on one of these drugs. I'm going to focus on propranolol and its pathway through the body, just to give you an idea of whether or not our method was successful or not. So propranolol was designed initially to treat hypertension. I believe 1950s was when it was first developed. It's good for heart medication. It's also good for stage fright if you get really nervous, like I do. And it's also been banned by the International Olympic Committee because rifle shooters and other sharpshooters will take it to slow down their heart rate enough so that they can shoot in between heartbeats to gain improved accuracy. This drug is metabolized in a couple different ways. That phase 1 metabolism can be seen here by what we would call the propranolol oxide, where we have an additional oxygen group on the end. And the propranolol glucuronide will be seen over here, where we have a glucose molecule that's been conjugated. That's the other type of metabolism I talked about. And we'll be looking at the concentration of all 3 of these compounds in a variety of different organs. Now, I had 10 organs originally, but I'm only going to show 4 just to sort of give you an idea of the narrative. Here is, for the record, just to show again, these are the 10 organs and the blood spots we had. We had 3 mice. All of them are dosed at, or 2 of them are dosed at 40 milligrams per kilogram, and then 1 is sacrificed at 30 minutes, so that should be really shortly after it was dosed, and then another one at 2 hours afterwards. And then we also have a control mouse with no drug at all, just to see a baseline level of how well our detection, what our signal to noise is, for some ideas. So here's the stomach. You'll note that on the y-axis here we're dealing with mean counts instead of a specific concentration, because the stomach right here sort of blew away our entire calibration curve. This sort of makes sense. You figure that they were force-feeding these mice propranolol into their stomachs, and then it's just sitting there, particularly at the 30-minute mark. What you see is a level that's incredibly high. And then at 2 hours, we do see a slight decrease in the concentration, but in the stomach, you don't see any level at all of the metabolites, and you don't see anything comparably in the control. Again, this seems like a pretty reasonable result at this point. There's a crap load of it. When you shove them full of propranolol, they have a lot of it in their stomachs at both 30 minutes and still 2 hours. Moving on to the brain, you'll notice first on the y-axis the magnitude. The order of magnitude now is 1,000 times less. We can see that propranolol is in the brain, and that by 2 hours its concentration has dropped by about half. And also that neither metabolite shows any appreciable concentration in the brain, at least as far as mean counts go. Moving on to the liver, concentration has now risen again to 20,000. Again, still several orders of magnitude less than the stomach, but beginning to show significantly higher concentrations. Propranolol is found, particularly at 30 minutes, but in particular we see that the glucuronide, the metabolite, that we'd be looking for to be processed in the liver is appearing quite a lot at both 30 minutes and 2 hours. Well, why there's a decrease of 2 hours exactly? I'm assuming when they choose 2 hours, again, I don't have the exact data on this, but the 2 hour mark, you should already be beginning to process it out of the body and moving on to clearing it out of your body. Which we can see from the kidney here, because the kidney has low concentrations of both propranolol and its oxide form, and it's the glucuronide form, what we would expect to be most excretable from the body. That is what we're actually seeing pulled out because it's in higher concentrations, not quite as high as in the liver, but still high concentrations. So hopefully this is a sign that we're seeing excretion of our drug. So putting all this together, we see propranolol absorbed in the stomach, distributed slightly into the brain, but not a whole lot, definitely not as metabolites. It's metabolized in the liver and excreted in the kidneys. These are like really normal, boring results. And that's exactly what the researchers we were working with wanted to see. They wanted to make sure that when they looked at what should be a really a well-understood drug, that our results look almost exactly what with a textbook result would look like. And that's because when we look at how we did this testing, we managed to test 2 drugs at 2 different time periods plus a control mouse, or sorry, 5 drugs at 2 time periods plus a control mouse. We did 10 different organs, plus we did blood spot sampling, and we did it, every single organ was in triplicate, and we did positive and negative ion modes simply because some ions were better, some drugs are better detected in a specific mode. And so that totals out to around 1,000 samples, which took a week once we finally got everything set up. So as far as the speed of how things were going compared to the previous method used, we were able to accelerate the process considerably over previous metabolic studies. So to conclude with some future directions, this work that I was able to conduct was only possible through funding from a lab from Novartis, which is a major pharmaceutical company from Switzerland. And they were very pleased with this data. And we were able, because we were able to confirm how efficient our technique was at speeding things along while still maintaining similar results, there's hope now that this process can be used for charting the metabolism of some new compounds and seeing what they can, what a new drug is capable of, how it's metabolized. So with that, I'd like to do a few acknowledgments quickly. I'd like to thank Dr. Dave, my advisor for the last 4 years, for all his excellent guidance. I'd like to thank Jerry Lokensgaard for giving me the opportunity to go down to Oak Ridge National Laboratory, which was a wonderful experience. I'd like to thank the entire chemistry department here at Lawrence for all their excellent years of teaching. I've learned so much. Gary and Vilmos definitely need thanking for their excellent guidance at the lab. Those guys really know what they're doing in terms of developing this technique and also showing it to others and sort of shouting it from the rooftop about how great it is. I'd finally like to end by thanking my parents for all their wonderful support for these last 4 years. And I'd like to thank all of you for coming here and listening. Thank you for watching. Also, Allison's going to be presenting in a few minutes. So if you all, I think it's at 4:15 or 4:30? 4:30. 4:30, okay, so 15 minutes from now there'll be another presentation on electrochemistry. Just announce that now, I guess. Anyone questions? Okay, Rachel.
I have a question about your Electrospray. So when you pick it up with the nozzle, you said you take a solvent and you put it down on top of your, whatever the organ is.
This
one.
you mean?
So you put the blue down on top of the red sample, and when you pick it up, it's red. So is it really difficult to pick a solvent that will pick up with the materials?
Yes, I didn't exactly mention this, but the laboratory that I worked at had spent a considerable amount of time calibrating a specific solvent for whatever analyte they were looking at, and what they found is that using 80% acetonitrile, 20% water is particularly effective at just a broad spectrum of picking up drugs. So one of the things is that it'll never pick up 100% of the drug on the spot. But one of the assumptions is that if we go to the blood spots, right? So if we build a calibration curve off of these known concentrations, if we measure, if we figure out what percentage of these is picked up, we can sort of transfer that idea to how much of, say, a specific organ or the drug in a specific organ is picked up and sort of infer, in a way, the concentration in that organ. Dan?
So the previous method with blender and chromatography and all that stuff, where That's an FDA-approved method for determining this? Or is
Oh,
that?
I don't know what the FDA approved method is.
Well, no, I mean, just like when you test a drug and show the data to the FDA and like, yes, you can move on to the next step. I don't know if the technique, my question was going to be, is the technique something that has to be approved by the FDA before you can even use that instead of doing the blender method?
Right. So my understanding is mass spectrometry is the tool of choice in both methods for measuring how much concentration you have, and what differs simply is how effectively you think it truly discerns the concentration within the organ. So with this former method, you take the whole organ and you measure the total amount of drug in that organ. That's a nice holistic approach. Spot sampling is perhaps maybe has some flaws in terms of if it's not evenly expressed within an organ, can you say that you got the same concentration everywhere? Not exactly certain, but we would assume that the cells pretty evenly express it. As far as that old method goes, it's tried and true. It's what they do. It's my understanding of this anyway. Joe?
So do you see any, or does the lab see any potential for using this method to help determine where drugs are localized within organs? So, what part of the brain is it most concentrated in? What part of the liver?
Actually, yes. One of the research publications they had, or posters, they had a few tissue cross sections of brain. I don't think it was not a human brain. I'm going to say that up front. But they were able to show that they could test specific regions of the brain and measure differential concentration levels of, say, a given drug. So that's one interesting application. Again, a mouse, if you go, so like an individual spot sampling in the brain would cover up something like 30% of it. So getting a differential selection. But you could use this method. They also developed this for all sorts of other things, right? So they could put a leaf and like freeze that to the slide and then measure throughout a plant or something. So the surface analysis works as long as you've got something thin enough that you think you're getting a good cross-section of cells. Jamie?
When you have to do the flash freezing, is there a potential that you're going to degrade the compounds you're looking at? And then also that when you have to cut them, are you going to lose sample because you're having to slice it?
So that's a very good question. I don't know. When we got these samples, they had been sent by the drug company, already sort of, they came as sort of a plastic film with a very, like the millimeter thick section attached to it already frozen. So I would assume that they were sending it on the assumption that they were losing very little sample, or else why would they have even considered beginning the process with flash freezing? So they had developed the freezing, the slicing, the mounting, all of that. What we were simply trying to show is that that was viable because we have the surface analysis technique that we no longer need to grind up the whole organ. Shannon?
Do you know about how long using the previous method, taking that many number of samples would have taken? Like, what's the sort of time?
No, I don't. I didn't have to do that method. But I can just tell you, for instance, adding a column to the work increases the time considerably. Ask any of the organic students. So, or you can think about, it also simply requires an increase in the amount of expertise you need. You need people who are, I don't know if they're lab techs or whatnot, but someone has to be the mouse surgeon to go nuts with all the organs and to do that all day long. So this is cutting out, I guess, manual labor, or cutting out, you need fewer people, so it should go faster. Ezra?
Do you believe there's a possibility that collectors from different organs with this liquid method could have multiple organ concentrations mixed together? Say if you got part of your droplet on multiple organs, including them.
Yes. See, that is actually the main problem with this technique. When we use our sample tips, they produce a droplet a certain number of millimeters wide in diameter. And the issue is that if you don't select points solely within a single organ, you could get, bleed over where you draw your solvent is drawing concentration from a sample next to it. So for instance, if you try and sample the spleen too close to the stomach, the whole thing goes off because the stomach has so much concentration that everything is thrown off. It just throws your spleen data out of whack. So it's possible to decrease the amount of solvent you use. That'll decrease the diameter. But yeah, at some point there's a finite limit to how small you can get in terms of your resolution. And that also means that how finite you can limit where you're sampling.
Other questions? Let's thank the speaker again.